All right, good afternoon, everyone. I'd like to get started for our Symposium 2. I would like to introduce our speaker, our plenary talk for starting Symposium 2. It will be my pleasure to welcome Dr. Bryan Newman. Dr. Bryan Newman is a lead pharmacologist and a team lead for the Inhalation and Nasal Drug Products team, which is in the Division of Therapeutic Performance 1 in the Office of Research and Standards in OGD at FDA. Dr. Newman's work has been focusing on developing product-specific guidances in addressing controlled correspondences, citizen petitions, consults, and pre- and meeting requests within this area. He also serves as a project officer and contracting officer's representative for the regulatory science research initiatives related to inhalation and nasal drug products. So he's very active in the research areas within nasal and inhalation products. Dr. Newman received his BS degree from Louisiana State University in biochemistry, and his MS and PhD degrees from the University of Michigan in pharmaceutical sciences. So with no further ado, I'd love to welcome Bryan Newman to the stage.
All right. Good afternoon, everyone, and thanks for that introduction, Dr. Bielski. So I'm pleased today to be able to open our discussion for the alternative approaches by going over some of the agency's understanding of these different types of studies, where we think these might be able to work, and also how we might want to design these across the different type of dosage forms. Here's the disclaimer. So for an outline, this talk will be broken up starting with a brief overview of the challenges with conducting the bioequivalence studies with the locally acting meteor dose inhalers and dry powder inhalers. This will then move to the design of alternative approaches for our solution-based MDIs and then on other complex inhalation products. I'll then start a discussion for the other factors that can impact an alternative approach design and then move to some conclusions and next steps. So when we work to develop a generic locally acting orally inhaled drug product, this obviously has a lot of challenges. This stems from the complexities of these products and some of the different sources I'm showing here. The patient-related complexity deals with the physiology and the challenges around measuring the drug at the site of action. Typically, this would be done using pharmacokinetic studies, but since those lie downstream of the site of action, you can have the uncertainty between whether when you're measuring the systemic concentration, whether or not that's actually reflecting the local drug concentration. The devices are obviously complex because they're drug device combination products, and they have a wide range of designs that can be used across the different types of dosage forms. There's also the different patient-device interactions that can present challenges and complexity, particularly for the patients being able to understand how to use a device, as well as adequately and appropriately administer the dose to them. For the formulation-related complexity, you can have the physicochemical properties of the API and the excipients, how these can interact with each other and impact performance, as well as the different types and amounts of inactive ingredients within the different formulations. So traditionally, our bioequivalence approach has utilized the weight of evidence approach that's been discussed today. It's consisted of the in vitro PK and comparative clinical endpoint and PD studies, and as well as the formulation and device similarity, with all of these being needed to be completed for you to establish bioequivalence. And as was discussed earlier, we have had success with these. We've had a few generics for MDIs and DPIs approved, but we also acknowledge that there's significant challenges with this approach. And of course, the challenges lie heavily with the comparative clinical endpoint study. So these studies, of course, use a number of patients, and that's ultimately where a lot of the challenge lies. They provide a higher level of variability and in turn lower the accuracy and reproducibility of these studies when establishing bioequivalence, and also the dose response issues with the dose lying on the flat portion of the exposure response curve, leading to lower sensitivity. So with these studies, again, they can necessitate, again, a large number of patients and a longer study duration, that of course adds to their cost and amount of time you have to conduct them. So again, it's important to understand if there's so many challenges with this, why do we include this type of study in the weight of evidence approach? So most of these treatments for respiratory diseases are going to be locally acting, treating their disease, their issue within the lungs. And on top of that, the FDA's regulations require the demonstration of a generic product and its RLD not having a significant difference in the rate and extent of the API reaching the site of action to establish bioequivalence. So, with that, we can turn to look at our in vitro BE studies and the PK studies as well, and understand that while those are included, there's also, some uncertainty with these types of studies as well. There's the uncertainty with the correlation between the in vitro performance and in vitro drug deposition. There's some, again, uncertainty with the relationship between the systemic PK and the local levels of the drug within the lungs, and ultimately the degree in which that these measures can sufficiently distinguish the differences in the clinical performance between a test and reference product that would impact their bioequivalence. So the main point is that, again, these clinical endpoint studies are incorporated because they can or have the ability to provide some level of assurance for local drug delivery equivalence between a test and reference product. So with that in mind, the next question is understanding what goes into the drug delivery process itself. So here I'm showing a number of steps or phases in the drug delivery process. And for a patient to adequately get the drug to their site of action, they have to go through a number of steps that I'm showing here. So your first type of step will be the formulation device characteristics that are going to influence the product. You'll have, after the patient actuates the device, you have your aerosolization of the dose. And then you also have, in step 3, like the formulations post-actuation. So this would be like the maturation steps that might occur. Following this, you'll have your airway transit and ultimately ending up in the deposition, dissolution, and absorption areas. And contributing factors could be the physicochemical properties of the formulation again, the device characteristics, how those are going to impact the dose administration, downstream aerosol properties, and even when you're depositing at the site of action, the microstructural characteristics of the particles impacting, like, say, dissolution. And then you can have your patient-related contributing factors. So again, how are they going to administer the dose correctly? Are they, the level of effort that they have their inhalation, the effort that they're going to use with their inhalation, is it going to be timed appropriately when they're administering the dose? So aside from just that general description of the steps for drug delivery, the next way we can look at this is what are the device-specific conditions or roles that go along when you're looking at this, because when you look at it through the lens of each specific device, we can see distinct differences in how they proceed through these different steps. The different MDIs will obviously have their APIs either in suspension or solution, whereas the DPIs will have a blended powder. And when these products are actuated, the MDIs will be propellant driven and utilize the evaporation processes to mature the different droplets to the eventual residual particles that form. Whereas with the DPI, the patient's inhalation is going to drive this and facilitate the deaggregation of the API from the carrier particles. And ultimately, across the different products, you can wind up with a different type of depositing particles. So you can have your dry residual particles or an API co-solvent droplet depositing, or you can also have an API or API carrier agglomerate particle depositing. So if we consider all of these factors, what we can conclude from the local drug delivery process is that the local delivery is a complex multi-step process. It involves each step needing to be completed and impacting the next. And the comparative clinical endpoint study is an attempt to evaluate each of these steps from the patient's interpretation all the way to the deposition and absorption when you're establishing local drug delivery equivalence. So when we look at what an alternative approach would be, The consideration is that it would be a combination of in vitro in silico or an alternative in vivo PK study that could account for these different steps or factors that impact the local drug delivery of the API to the site of action. These studies ideally will work together, and they'll provide a comprehensive evaluation of local drug delivery. In this case, again, in silico methods here would be or can be useful for demonstrating how the results from the different alternative approaches can work together to establish equivalence. Additional consideration with an alternative approach is that they would be able to provide an understanding of the relationship between systemic PK and the local levels of drug within the lungs, as well as inform on the correlation between the in vitro performance and in vivo drug deposition. And a last part that I'll add is that the types of alternative B studies may depend on the specific dosage form that you're working with and the type of formulation. So with this type of framework in mind, the next question to address is essentially, well, what types of studies are going to work here? How would we want to design this? So to address these types of questions and other questions, the FDA has utilized CDUFA-funded research to try to build a better understanding of how these products work, how the formulation and device interact with each other, how that impacts performance and quality, how that type of performance can then impact its clinical performance, and ultimately what types of new bioequivalent studies might be useful or sensitive to evaluate a test and reference product. So if we were to go back and look at the drug delivery process, the question would be, well, okay, what types of studies are going to be sensitive here, and what studies are feasible? So ideally, again, you can construct a method using a set of potential in vitro study methods, in vivo study methods, and in silico study methods. And here, again, I'm showing just a list of example or potential studies, as well as where some of these studies might inform on the drug delivery process. And one thing that you can, or one point that you can note here is that, again, this is a lot of studies that are listed, and the idea is that it's not necessarily do every study. The idea is to build a comprehensive alternative approach that's able to utilize a set group of studies that either address specific steps in the drug delivery process or work together to address certain steps in these processes, so that, again, the method is robust and comprehensive and can provide the same level of detail and assurance that the comparative clinical endpoint study is going to do. Naturally, the types of studies here might not all be applicable to certain dosage forms, so that's another consideration you'd want to have here. So with this in mind, when the FDA first designed their alternative approach with the solution-based MDIs, we started with this type of dosage form because it's less complex compared to the other types of dosage forms, and so we're able to make more conclusions here. And the general framework that's been presented is, if your formulation is Q1, Q2 the same, and you have a similar device, then a set of alternative studies could be used to substitute for the comparative clinical endpoint study. These would consist of your characterization of emitted sprays, your morphology and imaging comparisons, more predictive ABSD testing, dissolution, quantitative methods and modeling, and alternative PK/PD studies. So a question to ask is, again, why are these sets of studies actually selected here? So another way to view this is to look back at the drug delivery process and see how these actually fit. So if we start at the top, the characterization of emitted sprays is going to inform on your aerosol formation. Your morphology imaging comparison is going to inform on the formulation post-actuation. Your more predictive ABSD is giving you input on the transit through the airways and the deposition, and naturally dissolution is informing on the dissolution process. Now, these other studies at the bottom, they provide additional support, particularly because, for instance, the in silico methods can provide a wide range of support for across a number of studies, and can be used to help facilitate building the bridge between in vitro performance and the regional drug deposition. And likewise, you could utilize the alternative PK studies to help understand how the PK results correlate to local deposition. So with this understanding, the next step would be, okay, how are we going to apply this to the more complex products? So again, looking at the solution-based MDIs, we understand that for these products, the API is dissolved in the canister with propellants, co-solvents, other components, And there's multiple factors here that are going to be influencing the drug delivery process. So your formulation device interactions are going to impact your aerosol droplet formation. Volatile and non-volatile components would impact the maturation and evaporation steps, and your patient-derived factors would impact your regional deposition of the drug particles. So the recommended alternative approaches are going to evaluate these factors in the drug delivery process when determining if there's differences that would significantly impact the test and reference product's performance in local drug delivery. So the thinking here is that for other inhalation products, such as a suspension-based MDI or a DPI, these can have the same influencing factors, but they can also have additional factors that add to the product complexity. And it's this additional set of complexity that may necessitate additional information or studies be included in your alternative approach. So if we turn to the suspension MDIs first, we can understand that for this set of products, it's the suspended API particles within the canister that's providing the complexity and the potential impact on the drug delivery process. So how this can be come about is you have surface level interactions that can impact, again, downstream critical processes for local drug delivery, such as your van der Waal or electrostatic forces that can impact the API's particle size distribution over time through agglomeration. You can have surface roughness differences in the API particles that can impact stability, and the interaction of the API with formulation factors that are included to control for stability. These could lead to differences in the particle size distribution of the deposited particles. So what this would suggest is that for an alternative approach for a suspension-based MDI, a characterization of the formulation pre-actuation may be needed here. So with this understanding, if we were to look at, say, a hypothetical example of what an alternative approach would look like, first, we could look to see that a lot of the studies for the solution-based MDI can be included here, but with an addition of a characterization of the pre-actuation dose to understand the suspended API particles. However, we have to also understand what does this, by including this type of study, what does this mean in terms of the difficulty in meeting this alternative approach? So there's additional considerations here. For technical challenges, do we have a feasible method that can actually do this? Is there a sampling method that's able to maintain the API particles in suspension and provide this level of information? Is there a degree of correlation with the local drug delivery? So if you do the pre-actuation characterization, is this actually meaningful for local drug delivery? Does your device interaction impact this utility? And then can other post-actuation studies inform on the uncertainty that you're trying to evaluate when you're looking at this for the suspended API within the canister? And then finally, are there additional methods that you might want to consider for the rest of the alternative approach for a suspension-based MDI? So this presents some of the challenges and questions that we still have when looking at this and trying to determine what's the most appropriate alternative approach for this type of dosage form. When we turn to DPIs, we can understand that it's the blended API particles and the deaggregation process that's critical for the drug delivery process and how that can impact regional deposition and the rate and extent of absorption. Looking at the bulk formulation first, the API particles are typically agglomerated with themselves or with carrier or other excipient particles, and this agglomerated profile can depend on the physicochemical properties. So there's a number of conditions here that can influence this, and then those conditions can be impacted by the manufacturing process. So what this would suggest is that for an alternative approach for a DPI, again, the characterization of the pre-actuation dose might be needing to be included. And then following the actuation for if we look at the deaggregation of the API carrier particles, this is going to utilize energy generated from the patient's inhalation. Likewise, you have different internal geometries and methods of aerosolization for the different DPIs, and that can result in different resistance to airflow. And if we throw in the patients here, they can provide their own variable source of inhalation because the disease state might lead to patients having different abilities to inhale through the product. So what this would suggest is that a characterization of the airflow impacts on the post-actuation process would be critical too. So if we turn to a hypothetical example here, we could see that, again, many of the studies from the solution-based MDI could be included, but with the addition of the characterization of the pre-actuation dose, as well as some incorporation of the inhalation flow as part of the other study designs. But then as well, there are additional considerations. There's still the technical challenge here of understanding if there's a feasible method to evaluate the pre-actuation dose, making sure that there's a sampling method that can maintain the agglomerated state of the bulk powder, understanding the degree of correlation, local drug delivery. If you're going to do, again, the pre-actuation characterization, it's going to provide a meaningful local drug delivery. Understanding how the device interaction or the airflow impacts ultimately the utility of a pre-actuation characterization, and if other post-actuation studies can provide the level of understanding that you would want to get at by attempting to do this pre-actuation characterization. And then finally, you can then look to see, again, if there's additional methods that might be needed across the other study design. So again, these would be some of the challenges that you'd want to address for a DPI for an alternative approach. So to look at some of the potentials for newer methods that might be able to address some of the technical challenges, one of the methods here that I highlight is the X-ray computed tomography that is unique in its ability to offer a non-destructive characterization method that can look at the bulk formulation within the device or the capsule. And there's been publications to show that it can detect differences in lactose particles, from like larger lactose particles to lactose fines, or lactose particles and APIs as well. So there's certainly interest and potential here, but at the same time, there needs to be an understanding of the sensitivity and distinguishing the differences between a test and reference product using this type of technique, and ultimately how well it would inform on the local drug delivery process. If we were to turn to in silico methods, I'm showing here a few different methods. Of course, there's others that can perform here too, but just generally, in silico methods can provide a lot of information on the drug delivery process. If you look at regional drug deposition modeling, it can be useful for providing predictions for the regional drug deposition. If you look at computational fluid dynamics modeling, this can provide a better understanding of how realistic geometries within the patient or the device can impact performance, as well as maybe providing an understanding of particle-to-particle interactions. And then for physiologically based PK models, these can provide a way to predict local and systemic PK and better understand how the physiological processes like the clearance and metabolism can impact absorption and provide a level of understanding for those effects on the drug delivery process. Ultimately, again, these methods could demonstrate how the data from the different alternative approaches can work together to establish equivalence in local drug delivery. And then if we try to consider this specifically on what ways these an in silico method could address some of the technical challenges, again, for an MDI or DPI, we could consider, for instance, for the DPI, the in silico methods providing a way to characterize the particle-to-particle or particle-agglomerate interactions, how those affect the powder flowability and aerosolization, and the resulting drug and/or agglomerate regional deposition. For the suspension-based MDIs, likewise, it'd be a formulation under characterization to see how differences in the API particle size distribution would impact aerosol droplet formation and the maturation process. So again, the thinking is to utilize some of these in silico techniques to look specifically at the additional complexities that the suspension-based MDI and DPIs present, and try to utilize them to address those challenges by either doing this alone or in combination with some of the other alternative approach studies. Now, with that, I'll move to some other considerations that can impact your alternative approach design, and first turn back to the study population. So again, the comparative clinical endpoint study is conducted in patients, and so there can be a misunderstanding that because I'm doing in vitro studies or I'm not doing the comparative clinical endpoint study, then I don't need to consider the patient population anymore. Well, ideally, the patient population should be considered when you're designing your alternative approach, particularly for selecting the relevant study parameters. So you'd want to understand, where's your site of action? Is this, what portion of the lungs or the airways is the drug affecting, in that case, understanding the API's mechanism of action and where it's depositing, as well as looking at the airflow rates and understanding if you're going to consider which breathing profiles to choose, does it make sense to utilize a healthy subject's lung inhalation rates when you're trying to characterize deposition in a diseased lung. Now, if we turn to the study purpose and why you're actually conducting the study, we know, again, you're going to do an alternative approach to inform on a drug delivery process, but how you do the method development, how you optimize it can take different directions. And so you can go into a more sensitive or discriminatory focused design, or alternatively, you could go in the opposite direction. You can go into something that's more biorelevant. And ultimately, the direction you go could, again, depend on how you plan on using the study. Is it going to be a standalone method that's only evaluating just one specific process in the drug delivery process? Or is it going to later inform on, say, like an in silico approach that would be better informed if the parameter values you're providing it are from something that's more biorelevant? And then finally, for the APIs, API properties themselves can present challenges with developing an alternative approach. And an easy one to understand is the high solubility versus low solubility case. If you're to look at dissolution studies for a high solubility API, this may present limited discriminatory capability. It may just present a difficult challenge to develop your method. In contrast to when you're working with the lower solubility API that may have better utility, better relevance coming out of this study for that particular API. And of course, there could be other studies specifically like a charcoal block study that are more suited for one set of API versus another. And then finally, for the formulations with more than one API, overall, your approach is going to want to be considered and comprehensive for both of the APIs. You're going to want to not necessarily have to have every study applicable to both APIs, but that the alternative approach consisting of whichever studies for that one specific API is going to be sufficient. So for some general conclusions and next steps here, we can conclude now is that, again, the comparative clinical endpoint study presents challenges to establishing local drug delivery equivalence for these types of products, and the agency's research efforts have presented alternative approaches that we've utilized for several solution-based MDIs to address some of the challenges of having to do this study. Again, these studies can be a combination of the different in vitro, in vivo, or in silico studies, and these would evaluate your key steps and contributing factors to the drug delivery process. They'd also address the relationship between systemic PK to local drug levels within the lung and inform on the degree of correlation between your in vitro product performance and local lung deposition. Using this understanding, if we turn to suspension-based MDIs and DPIs, we can see that there are still remaining challenges here with these, and they require additional consideration because of their additional complexity. So because of this complexity, that may warrant additional information on studies being included in their approach, but this needs to be weighed against the study method considerations that I discussed earlier, how the device is going to influence the impact of that type of study, and then potentially what other sources of information may be useful for addressing the complexities you're trying to understand. In silico approaches, again, here can help facilitate this and may provide additional options. And looking at other product-specific factors, again, your study population, API's mechanism of action and site of action can be critical for how you design your method, the specific purpose of the study method, and the overall alternative approach, and also the API's physicochemical properties and other formulation factors. So where do we go from here? Addressing the remaining challenges with identifying alternative approaches for suspension-based MDIs and DPIs requires additional discussion and input from industry and academia on particularly determining the appropriateness of currently available methods to meet these challenges, as well as identifying where there could be potential knowledge gaps that would necessitate additional research. So with this, the next stage of our workshop here is the following two sessions, where the first session is going to be focused on in vitro bioequivalence methods and understanding how those work for the alternative approach. And session 2 is the in vivo and in silico study approaches and how those fit into alternative approaches. So so these sessions will be going on at the same time again, as been described. So as you attend these sessions, try to consider the questions that I've raised here. Try to think to yourself how you would be designing your alternative approach, how, whether or not you have confidence in certain methods over others, or whether or not you need more information. And hopefully we'll have a good discussion with the rest of the workshop. So with that, I'd like to acknowledge all the individuals here, particularly our external research collaborators that gave us a lot of the support for where we are now. And for any questions and such, please provide those in the chat box so we can keep a good record of them, and then potentially address some of these in the panel discussions later in the workshop. Thank you very much.